Hi everyone, this is Jamie. I'm a pharmacology PhD student at Stony Brook University, and today I would like to talk about my project, also collaborating with Donald, about the damage provoked by the exposure of the lunar regolith stimulant to human lung cells. So first of all, as John has already mentioned, so lunar dust can be really tricky to deal with based on like a lot of previous report. I'm not going to repeat that here because of the time. And although it's somewhat similar to what it's made of with the terrestrial dust, but it can be more toxic and reactive. So and as John has also said, it's exposed to space radiation, solar wind, and other stuff. Um, so that's why it becomes so reactive. And there are a lot of like astronaut in the past talking about this also. So that's why we would like to look into this kind of issue. So based on our previous research, based on the paper we have published in 2018, we hypothesized that after occupational exposure to lunar dust simulant, lunar dust, those lunar dust will lead to DNA mutations, no matter in nucleus in the cells or mitochondria in the cells, which this organelle is responsible for the energy production to maintain the survival of the cells. But the nuclear DNA and the mitochondrial is kind of somewhat related to each other, so they cannot live without each other. Once mitochondrial has some dysfunctioning happening there, it will also lead to some problems with the nuclear DNA. So with this kind of phenomenon happening, it'll lead to further tissue damage and elevated cancer risks, such as lung cancer or other part of the tissue that may be exposed to lunar dust. So as Donald has also said, we will basically, we got the samples from the NESFELS lab, and we will do a size exclusion and grind it up to around 10 micrometer. And for some of the samples, we'll treat it with hydrogen gas at 900 degrees Celsius to simulate the, the solar wind on moon. So we'll just call it for reduced material, and we'll also like compare the differences later and when I talk about my experiment. And basically for all the time, we'll store it in vacuum desiccator. And before the experiment, we'll grind it up again to make sure that the side is not oxidized. And then we'll directly apply to the human lung cells. So here's the breakdown for the two lunar dust tubes. It's what Donald just mentioned as well. So first of all, I would like to give everyone an overview. So basically, after the lunar dust was like applied to the cells, we can see a significant differences here. Like the cell viability was like strongly decreased, with the dose dependency, as John also mentioned, I guess, I believe. But for the reduced materials, when cells are confronting them, it can be like worse. Like compared to the untreated one, it can be worse. And it's also happening, there's also dose dependency for the reduced samples, although they are quite low here. Let me see. Okay. And next, after looking at like a big picture of what's happening there, we would like to see if the mitochondria is like facing any issues. Are the function impaired? That will affect something else. So we would like to give everybody an overview with some images. So with the untreated control, there's nothing added. You can basically see nothing in the mitosox rat dye, which is a probe that is good for detecting the superoxide inside the mitochondria. So you can see here after treated with some positive control, there's a round, there's some like round stuff forming here, which is the mitochondrial complexes being stained. So you can see that's happening. And we can also see some of them after they're treated with some of our lunar dust simulants. And when the cells are under really high damages, you can see some of the cells are deformed and they're just not looking good. The nucleus are also stained. That means like the cells are under severe damage. It's really bad. And then we quantify it with the plate reader, and we also found a dose dependency, which is not listed here because of the time limit. And we also found out that the reduced samples are also showing like much higher superoxide levels inside the mitochondria. Okay. And next, we would like to see if the mitochondrial DNA is also affected, because that's also responsible for encoding some of the proteins that's important for the mitochondrial function. So we use a PCR assay to do so. And we have done 2 PCR, which will probe the shorter fragment and the longer fragment, and we will use the ratio to normalize our results. So when we see like a lower ratio, which means there's more DNA damage there. And our results also show the same thing, that after the exposure to the reduced samples, you can see a much more like dramatic, much more apparent decrease in the level in the DNA damage here. So basically, it's showing the similar stuff, like the cells are under serious damage. The mitochondrial function is like extremely impaired. And we also like to see that for what we haven't seen before, we haven't published it, is that if the nuclear DNA is also under serious damage. So we use an assay called alkaline combat assay, which allows us to lyse the cells and then run an electrophoresis and check on if the cells are if the nuclear DNA is under damage. And you can see here in the control, they're looking pretty intact, and you can see the round shape here. But when we increase when we expose it to different simulants, you can see like there are tails forming, which indicates that it's under like serious damage. So I think I have to wrap up. So for all the results I have shown, both simulants, including LMS1 and LHS1, they both cause dose-dependent cytotoxicity, mitochondrial function, which is the superoxide level, and the genotoxicity, which includes the assay I just told you, and also the assay we use to detect the mitochondrial DNA levels. And the reduction of both materials leading to enhanced toxicity. So you can basically see that apparent effect easily with all these assays. And we are also interested in detecting what some other minerals that's composed of the Lunar simulant we're currently using, and we would like to see like what kind of minerals may be the main toxic components to cells. And lastly, although like we see a lot of damaging effect here, but we're not sure like physically or chemically what exactly things are happening, because in our previous published literature, we didn't really see the correlation that Donald has found, like with some reactivity effect, it just didn't match with the cell response. So we guess there's something still happening there. We haven't found it. And we also are working on doing in vivo experiment, as John has said, with the rats, with our collaborator. I hope he's here, Greg. I'm not sure if he's here, and to validate if our in vitro result is like really real. So that's all for my presentations. Thanks a lot for listening. I'm happy to take any questions.
Excellent. Thank you, Jamie. So if anybody has questions, please, you can also just raise your hand or unmute yourself and speak verbally, because we are going through the discussion session also. Or also include the questions on the chat.
Yeah, I guess I don't mind, yeah.
So yeah, go ahead.
Sorry, this is Elizabeth. I realized I forgot to raise my hand. Yeah, and I'm getting some answers to my question in the chat as well. But I guess my general question, like for this session, was like, is there any way to, like, you know, relate like the toxicity that you're seeing with how someone's immune system might respond? Just because it seems like there's a lot of variability in people's immune response to things. Like I have issues with my own immune system, so like I probably read more about this than I should, but like also I'm thinking of like even with like COVID-19, it's like, you know, some of the damage is from like the actual damage to the cells, but then it's like, you know, what the immune system does with that can like make or break the situation. So I'm just curious about how you might account for that.
So basically, I will say probably this one will be a better move because like there are a lot of like immunity is a lot like related to the kind of the superoxide. I think John has also mentioned that like the superoxide levels and the ROS inside the cells. So when like there's the stuff, for example, the Lunar dust stimulant, when astronaut breathe them in, and it'll like it'll lead to like a lot of generation of the ROS that the astronaut that the cells cannot really deal with it, then this will lead to some immune response. I believe so, but I'm not an immune person, so but like from what I learned, that's all I can tell you. Yeah, that's a good question though.
Yeah, no, thank
We
you.
may need to look into more like cytokines and stuff to like confirm like how highly it's related to the immunity. But yeah, that's a good point. Yeah.